Hematopoietic stem cells has been of interest of us, especially Dr. Yoko is a long-term collaborator. She is the person really pioneered this concept in that setting. So the bone marrow transplantation, I don't have to tell you exactly its importance. The previous speakers, they already talk about this. But our purpose is to use bone marrow as a means of rebuilding the immune system for the purpose of inducing tolerance to foreign tissues. So that's our concept. And the work has been done, a long time back. If you have mixed chimerism, David Sachs and Ilstad in early 80s demonstrated that indeed mixed chimeric recipients will accept not only allo but also xenograft. So it's a very powerful means of inducing tolerance because it goes through what they call central deletion as well as peripheral immunoregulation. Pretty complicated. Now, what are the problems? These are three major problems in hematopoietic stem cell transplantation. The first one is being stem cell competition. You have to have a niche, and the way it is done clinically, you precondition the patients with chemotherapeutic agents as well as irradiation, etc., so that you open the niche for the stem cell that you're going to put in. And engraftment is certainly a problem. And then you are now faced with graft versus host if you have mature T cells in the bone marrow inoculum that you put in. And interestingly enough, the T cells are required for efficient engraftment. And still we don't know exactly how they do that, but certainly that's what it is. If you have stem cells alone, there is an issue for engraftment. And those T cells, once they enter a recipient, because they see the recipient antigens as foreign, they get activated, they attack the recipient. That's your graft versus host. And the opposite, of course, the host responds to the bone marrow as a foreign body. So we have to overcome all these three. And presently, as I mentioned earlier, you precondition the recipient in the clinic, put hematopoietic stem cells or complete bone marrow. Of course, immunosuppression is on board, etc. But I must admit, fortunately for the community as well as for the patient, there are recent work by several groups in this country, especially where I am right now at the University of Louisville, Susan Ilstad, demonstrating that indeed it is possible to achieve tolerance. This is done in the context of kidney transplantation. I think that data is very exciting. So our approach, can we use our concept basically to deal with these two aspects, these two limitations of bone marrow, that is graft versus host and host versus graft. And so we used SA fast ligand. This is a chimeric protein. Here is the technology, just to remind you again. And this is the model. So the model, we used C57 black 6 to BAL-C. Now, actually, we've also done the opposite. If the same thing is true. 30 million bone marrow cells engineered with fast ligand. And then we use irradiation, 300 centigrade, followed by 10 daily doses of rapamycin as an immunosuppressive and immunomodulatory drug. As I mentioned earlier, it has synergy with fast ligand. I'm not going to go into it. And then we look at chimerism. So here is to show that, yes, bone marrow cells, you can put fast ligand on there. This is biotin, and this is streptoavidin as a control protein, and this is fast ligand on the surface of bone marrow. If you notice, everybody has fast ligand on their surface. And then when we do the transplantation, you follow the animals, for donor chimerism. If you notice, the group that have received fast ligand and on their surface, they are chimeric, but not the control group, the bone marrow cells with streptoavidin and rapamycin. And then at the end, experimental endpoint, when those animals were analyzed, there is multi-lineage chimerism in these animals. So fast ligand, it prevents the rejection in this setting, and it establishes multi-chimerism. One thing is important in this context is that fast ligand has tropic effect on stem cells. Somewhat differentiated stem cells, they express fast and they're sensitive to fast ligand-mediated killing, but not undifferentiated, truly hematopoietic stem cells. So that is something Nadir Askanasi actually published this data. It also helps basically engraftment. We don't know how that happens, but it's something we're looking into it. And then, if you go ahead and do the transplantation on this animal, this is skin transplant, and all of these animals except one, they did not reject the skin coming from donor, but they rejected the third-party skins, which has been shown by many others. So in this configuration, fast ligand prevents the rejection. Now the problem with this model is that the second issue is graft versus host disease. In rodent model, you don't see graft versus host disease that you see, for example, in non-human primates or in humans. So we wanted to establish a model where we see graft versus host disease and ask the question whether fast ligand can prevent graft versus host disease. And the reason we think it will, and this is based on, again, this cartoon right here, T cells, when they're naive, they don't express fast, they get activated, upregulate fast, as well as fast ligand. And upon repeated encounter with antigen, they become sensitive to fast ligand-mediated killing, either on the same cells or different cells, and you eliminate them. So we thought if we put fast ligand on the surface of T cells, mature T cells, when they get activated, they should express fast on the surface. Therefore, they should be sensitive to actually fast ligand-mediated killing. So conceptually, it makes a lot of sense. And it doesn't really happen, and here is the model. So take bone marrow cells. This is, again, C57 black 6, or you take splenocytes, you do your engineering, and simply you take these cells. We use F1, 57 black 6 by biopsy, so these animals, clearly they're tolerant, to 57 black 6. They will not respond, to the graft, but the graft, the mature T cells in the graft should respond and kill these animals, and that's exactly what happens. So again, showing that you put fast ligand on the surface of both bone marrow cell as well as splenocytes, and then when we look at apoptosis in vitro, if you notice, cells that receive fast ligand, they undergo apoptosis. Okay, and this is the control, and these are primarily, T cells. They get activated. This is in vitro mixed lymphocyte reactions. You take those cells, you look at them. They undergo apoptosis, and we've shown in vivo actually the same thing happens. Now, when you transplant the bone marrow cells with splenocytes in different configuration, you have irradiated control. Everybody basically expires from acute graft versus host disease. Bone marrow, splenocytes, the same thing. Bone marrow, and showing that, yes, this model developed actually graft versus host disease, okay? Because you have splenocytes and you have bone marrow. Bone marrow should rescue. We don't have that control here. And then you have bone marrow, splenocytes with streptoavidin. Again, those animals expired from graft versus host disease. The only group they don't expire from graft versus host disease, those are the group they received splenocyte engineered fast ligand engineered splenocytes or double engineering. So in both configurations, this regimen actually working very nicely. Okay, so if you look at the body weight, and again from body weight, these are the only group they basically maintain a study body weight, and this is. Looking at basically scoring the graft versus host disease. Again, these are the only two groups they did fine. Okay, this is again, this is just show you some picture. This is the control group, and these are the animals they've received fast ligand, so they're clearly looking like normal animals. And so we're now trying to further, work out, this regimen. So the idea from our perspective is that can we get down maybe to low level of irradiation in patients, clearly 200 centigrade is being used clinically, approved by FDA, but we're hoping maybe we can improve it further so that we don't we don't need to use irradiation. So we're playing around with some concept, as I mentioned earlier to you, is that our advantage is that not only one molecule, we can put 2 or 3 molecules with synergistic action, and indeed we've done it with another molecule. I'm not going to disclose it here, but in that configuration, it is extremely potent. You get beautiful engraftment at lower, irradiation. So the idea can really get rid of irradiation. So with that, it seems to be it's pretty effective and clinically clearly conducive, approach, because you're not really doing much. The whole process is pretty straightforward. And with that, I appreciate the people that actually did the work. Again, Nadir Askanasi is a long-term colleague. He did most of the bone marrow with Esma Yorku, the funding source from NIH, American Heart, and others. Thank you.